Hello everyone, welcome to our poster. My name is Jianhao Peng. I'm a PhD student from the University of Illinois at Urbana-Champaign. Today I will be presenting our work, SimiC enables the inference of complex gene regulatory dynamics across cell phenotypes. This is a joint work with my advisors and collaborators from SimiC universities of Navarra, Spain, and my colleagues in universities of Illinois. As most of us know, compared to bulk RNA-seq data, single-cell RNA-seq has the potential to reveal complex regulatory interactions across different cell types. However, most existing methods for single-cell gRNA inferences are inherently designed for bulk data analysis. Hence, they only produce one gRN per input datasets. The motivations of our works contains 2 points. First, we believe that related cell types should be characterized by similar but distinct enough gRNs. And second, differences in gRN and gene expressions can capture the changes of the undergoing cellular function shift. So with that in mind, and to overcome the limitations, we propose SimiC. SimiC is a gRN inference algorithm for single-cell RNA-seq data analysis. It requires inputs single-cell expression data, a list of driver genes, and the labels of the cells. SimiC will jointly learn gRNs with added similarity constraints across contiguous cell phenotypes. It also provides a framework to evaluate network dynamics through Regulon activity score defined per driver genes. We also proposed Regulon dissimilarities of each driver genes across types for clusterings or downstream analysis. The main body of SimiC contains 2 parts. Part 1 is the gRN inferences for different phenotypes. So we have the input here different 2 phenotypes single-cell RNA-seq datasets. And the main optimizations of SimiC is a fused lasso with square error terms of contiguous gRNs as the similarity constraints weighted by lambda 2. After the algorithm finds the optimum weights for different phenotypes, it will produce an incidence matrices between the driver genes and targets as the gRNs for different types. So we have W1 and W2 here, where the columns are target genes and the rows are driver genes. By comparing the per rows or per columns, we can see the differences but similar gRNs in these 2 types. The second part of SimiC is Regulon activity score computations. Once we have the incidence matrices from the previous optimizations, we can sort it the weight for one driver genes, or we can sort it the weights of the associated targets of one driver genes by the target's expressions, and then computed the Regulon activity scores as the area under the curves of the accumulated Ws. The Regulon activity scores is a score between 0 and 1, and the larger it is, the more impact the weight has on the target's expressions. We can color-code the cells by one driver gene's Regulon activity scores, and the picture here shows the activity levels of these groups of cells is much higher compared to the remaining. Once the Regulon activity scores per driver genes are computed for all cells, we can compute the distributions of Regulon activity scores in phenotype 1 and 2. And as you can see here, we define the Regulon dissimilarities as the total variations of the distributions in phenotype 1 and 2. If we repeated that for all driver genes, we will have a vectors between these 2 types as shown in here. And if we do it for all different cell types, we will have a heat map showing the matrix of the Regulon dissimilarities. And these we can use it for clusterings between genes or clusterings between cells. We apply SimiC to 2 real single-cell datasets. The first one is a longitudinal single-cell RNA-seq dataset generated from non-Hodgkin lymphoma patients treated with CAR T cells. SimiC inferred 2 gRNs from 2 time points. One is the infusion products IP and 12 days after the tumor recognitions. We recognize 3 cell states in both time points, and the example here shows one of the Regulon activity scores for the driver genes MYC, which is a proliferation marker. From the pictures here, we can see that it has the highest Regulon activities on memory cells and gradually become effector lives when it goes to next stage. When we look at opposite genes, RUNX3, it shows the opposite progressively increasing activities, so it becomes more effective lives in these two time points. And we can see that in exhausting stage, both Regulon's activity diminishes as expected. The second dataset we applied to is a dataset of regenerating liver after partial hepatocyte obtaining samples before and 24, 48, and 96 hours after the surgeries. From the Regulon activity scores distributions, we recognized two markers. One is the CEBPB, which is shows the initiation progression patterns. It's captured by SimiC and show the pseudotemporal transitions at single cell levels. So you can see that it has the highest activities at 24 hours after surgeries, and it gradually become inactive when before the surgeries or 2 days after the surgeries. An opposite example is CEBPA, which is the termination rematurations patterns. It shows exactly the opposite behaviors in and the reverse transition activities. We also use the Regulon activity scores for the clusterings, and we compare it to some of the existing methods, including SimiC and ICA-NET and CellRat. You can see that SimiC shows better or matching clustering performance when compared to these methods. Okay, thank you. And if you have further questions, I will be around the posters, and you can also have a look at our abstracts. Thanks.